Again, thank you, Mr. Santos, for sharing your insights. Even though the IVPT tool has been utilized in the field of research for decades, the IVPT method is relatively new to demonstrate bioequivalence for topical products in ANDA submissions, and there are a lot of hows and Ws regarding the conduct of this type of studies. During the discussion within the organizing committee members that include the representative from the agency, CROs, industries, apparatus manufacturers, and consultants serving in this area, we identified series of commonly occurring questions for the same. We already heard a historical background about IVPT study from Dr. Mabek, followed by Dr. Rene, about the current thinking on IVPT studies for ANDA submission. In this session, I would like to discuss point by point on the same topics. As I mentioned, I would like to request you to leave your question and comments to all panelists. So this is the disclaimer. This presentation reflects the views of the authors and should not be construed to represent FDA's views or policies. Now, I would like to introduce the panelists for this session in the alphabetical order of their first name, who had extensive experience in the field of topical and transdermal products. In the interest of time, I will give you a very brief introduction to our panelists, and you can find the detailed bio on the website for this workshop. We have Dr. Chudri from Division of Bioequivalence 3 within OGD FDA. Prior to FDA, he worked as senior scientist at an early stage biotech company, and later he worked as a principal scientist at Turgis Pharma. We have Dr. Audra Stinchcomb. She is currently a professor at University of Maryland. She is also the chief scientific officer and co-founder of F6 Pharma, a dermal product development company. We have Dr. John Lane, is a chief scientific officer at MedPharm. He has over 19 years of experience in the pharmaceutical industry developing topical and transdermal medications for early development to the post-marketing support. We already heard a nice presentation from Mr. Santos. We have Dr. Narasimha Murthy. He's the founder and director of the non-profit research organization that is Institute for Drug Delivery and Biomedical Research in India. He's also the chief scientific officer of the CRO based in Mississippi. We have Mr. Paul Lemon. He's the Vice President and Head of Dermal and Transdermal Research at QPS. Mr. Lemon is a recognized expert on in vitro and in vivo percutaneous absorption pharmacokinetics, dermal and transdermal bioavailability, and bioequivalence. We have Dr. Rene, is the Associate Director for Science at ORS and serves as the Chief Scientific Advisor for Topical Product Bioequivalence. Dr. Rene has been a principal or sub-investigator on over 400 pharmaceutical product studies and has held senior management roles in industry and serves on multiple expert committees and panels for the US Pharmacopoeia. Last but not the least, we have Dr. Nelamathu. He is the chairman and CEO of Turgis Pharma. His efforts over the past 26 years in various dermatological companies have led to many commercial products in the market today. Without any further delays, I would like to begin our discussion. I would like to start with consideration for the apparatus. When it comes to apparatus for IVPT studies, there are no recommendations about compendial apparatus unlike IVRT studies. Now, earlier we heard about, the most commonly used apparatus being a vertical diffusion cell, a flow-through diffusion cell, and there's some other. The first question that arises is, what are the different types of apparatus that can be potentially used to conduct IVPT study? And you can enlist the name of apparatus that you routinely use to conduct the studies.
I'm going to start with a little commentary from our side at Turgis. I think for most of the audience, this is a discussion amongst the panelists trying to address your questions. So most of the information you may have heard through presentations by Dr. Santos, Dr. Rane, and many others. So some of the information could be repetitive, but us trying to discuss amongst us, trying to provoke thoughts amongst you and also clarify. So with that said, the two usual suspects are always going to be vertical diffusion cells or flow through. And conventionally, for most of the generic products, we've been using vertical diffusion cells. And for new chemical entities with limited knowledge or limited solubility data, etc., we try to use flow-through cells. Again, within diffusion cells of vertical nature, there's various diameters and various applications depending on the dosage form. Any other comments?
Yeah, I think one issue is comparison of studies between labs using different kinds of diffusion cells. Flow through and a static are designed to produce the same type of result, but there may be sufficient subtle differences where the time to peak flux, for example, will vary between the chamber designs. So it may be an issue about lab-to-lab variability is really lab-to-lab comparison in equipment used.
Yeah, I'd have to second Virginia as well. I think, MedFarm, certainly we have quite a few of them, and our selection involves, depending on the study, so NCEs versus bioequivalents. We actually do some initial comparison analysis during method development to help us select out, because what we're finding is with certain products, some of the advantages or disadvantages within the cell design may work into your favor. That could be simply detection limits because of the lipophilicity or the low concentration of the drug, but it could also be a dilution effect with flow-through cells. So we actually run a comparison in the beginning to kind of help identify which diffusion cell or the size of the diffusion cell that we select in the beginning.
We have been working on FDA-funded project past 7 years, and we mostly use the Franz diffusion cells, the static vertical diffusion cells, for most of the experiments that we did. And in one of the experiments, we compared the flow-through apparatus with the static Franz diffusion cell. Of course, there was difference in the flux profile for the same product, between the two apparatus, but the end result, in terms of bioequivalence of a generic product with the reference product and non-bioequivalence of a product that was significantly different in terms of microstructure, was similar, in case of both the apparatus. So our experience, from our experience, what I can say is, regardless of the equipment that you use, if the IVPT method or the protocol is executed appropriately, I think the end results and the interpretation would be same.
Yeah, I think I wanted to comment on what Dr. Lehman said too. And what Dr. Yang found in her experiments with the flow-through cells. You do have some tubing that you have to account for, so we end up just extracting that lag time of the sample getting through the tubing, and we just add that lag time onto the time scale of the data.
So thank you everyone for your feedback. And what I'm hearing from the group is, there are multiple choices that we have to select the IVPT studies. And a majority of the time, the vertical diffusion cell is a starting point, and then really depends on the drug product. You need to change the apparatus. Any comments from the FDA folks on the selection of apparatus?
Yeah, so I don't know that the vertical diffusion cell is necessarily the starting point. It may be that you have experience with the flow-through diffusion cell and you start right with that. Dr. Murthy, I wonder if you could actually turn your camera on as well. We worked with Dr. Murthy, and we also worked with other research collaborators, Dr. Mike Roberts and Yusuf Muhammad, and we actually did some studies to compare and to try to understand why would there be differences in the flux profile created by differences in the design of the diffusion cell. And one element relating to the way that you design these comparative studies is to really kind of understand what is the effective sampling rate. And if you can start to actually calibrate your effective sampling rate, in other words, how much sample volume is being replenished over a given period of time, you start to get profiles that really align reasonably well between the different apparatus.
Thank you, Dr. Rennie. So we already touched base about the how to select an appropriate status. It really depends on the drug product and the investigation. Moving to the next question is, in the draft product specific guidance or PSU of acyclovir, there are details about the selection of apparatus in the method development and qualification of the apparatus in method validation sections. Would you please share your understanding about selection of apparatus versus qualification of apparatus?
Of course, I think they go hand in hand between the selection and the qualification. Like Sam and many others said, once you start with something, stick with that. If you start with a flow through or a vertical, but within the vertical, for example, you have a few other nuances, right? And depending on the dosing volume and the diameter of the vertical diffusion cell, as well as the nature of the pharmacoeconomics too. If all things being equal, if the study can be developed with smaller diameter, smaller dosing volume, some of the RLDs are very expensive. So when you're trying to optimize, choose your cell wisely. And once you've done that, the qualification comes along with it. Like Audra said, there's a tubing involved for flow through cells, and then there's absorption of the drug sometimes, particularly new chemical entities where we don't know enough about the molecule. So depending on the choice of operators you choose, the qualification goes along with it. How much of the volume that you are losing in the tubing, and then how to detect the lag time and qualify that particular apparatus.
Yeah, it's been very easy for us to work with Permagear and make modifications to our diffusion cells. And usually we just have to make a modification to the cap or some kind of a support system or some other type of replacement. And so we can modify to make smaller or larger surface areas for testing.
Not only in discussion of the cell, but the apparatus around the cell. Control of the water bath temperature or the heating block temperature and consistency over time, stir rate, also components that may influence the rate and extent of absorption.
Yeah, I would key off that to say that control element is a key part. Can you perform a well-controlled study? If your diffusion cell has volumes that are quite different from diffusion cell to diffusion cell because they're hand-blown glass, well, then you need to qualify that to understand what impact that's going to have and then account for that in the calculations. Ideally, and typically, you can have the dosing area be quite consistent, but you need to make sure that you kind of qualify and understand that. If you have tubing involved and the length of that tubing isn't precisely cut for all your diffusion cells, and you have different lengths randomly of tubing, then the lag time is going to be all different. And so the apparent time point they're taking is going to be all messed up. So part of the qualification process is to make sure that you understand the impact of the variations or control parameters of the apparatus and how that's going to impact your data, control all the things that you can, account for the things like cell volumes that need to appropriate in the calculations. I think that's the part that goes into the qualification of apparatus, and often that can be done when the apparatus is first brought in and the individual diffusion cells qualify the apparatus, and then it's qualified and you can use it consistently in IVPT studies thereafter. Whereas the selection of the apparatus, I think that has more to do with the specific method that you're working with. You might say, well, I'm going to start with a vertical diffusion cell and find, this is an extremely hydrophobic compound. I'm not getting great data here. Let me try a flow-through diffusion cell because there are some technical advantages that I think I can gain. And then you select the apparatus differently from the qualifying it. You select it for that method based upon kind of the scientific principles of what's going on in your study.
Thank you everyone for your input. And what I'm hearing from the group is, even though the qualification of the apparatus is listed as a validation parameters, it's independent of the drug product you are working. All the apparatus, should be qualified at certain time period, and selection of apparatus is then we are talking about specific drug product. So we have one incoming question, asking that how frequently the diffusion should be qualified, would it be appropriately to qualify annually, semi-annually, or before every GMP test? Any thoughts from the group?
I mean, I'll speak up here. I mean, we typically do those qualifications annually because they can be quite labor intensive, especially if it's the static vertical diffusion cells where you have to measure things and then weigh them out. And if you have hundreds, that can be time consuming. It's also unlikely that those parameters are going to change very much given the fact that they're glass, so you don't expect big changes. I will say that things that we, like for instance, in the flow through cells, tubing, because those are moving parts, so there can be some stretching involved. Those tend to get qualified on a different schedule. So I have a presentation on Friday where I talk about the different diffusion cells, and one of the things I keep harping on is just get to know them. None of them are perfect. All of them have limitations. Just understand the limitations, and then some of those limitations will help you define how frequent you qualify different components of the apparatus.
Thank you, Dr. Lane. And as you just said that we have a whole dedicated session about diffusion cell apparatus on a day 3. So let's spend not too much time on the apparatus as we have many other things to discuss for the IVPT studies.